In this video, we're going to be going over a brief introduction into metabolism and bioenergetics. All right, so to start off with some basic metabolic terminology, the term metabolism that you hear people say all the time, what it really means is the sum of all the chemical reactions in the body. So it's not just the energy producing chemical reactions or the energy breaking down chemical reactions like we often think of. All right, so metabolism can be divided into anabolic reactions and catabolic reactions. Anabolic reactions are the ones that synthesize or make a molecule so basically bringing two molecules or two more molecules together into a single molecule. That would be anabolic, where catabolic reactions would be taking a single molecule and breaking it down into more molecules, so smaller, simpler molecules. And then the term bioenergetics, which is really what we're trying to get after in this chapter set, is actually what most people mean when they say metabolism. So they mean the conversion of foodstuffs into energy that the body can then use for different processes like movements and exercise and lots of other things. All right, so by foodstuffs, I'm talking specifically the energy containing macronutrients, so the carbohydrates, the fats, the proteins, and also when it's present, alcohol can contribute to energy production. Of course, it's not recommended to do that, but it can be done. So when I talk about energy, what I'm actually talking about is a molecule called adenosine triphosphate, or ATP. And this is the basic unit of energy that the body uses. And it's the energy molecule that powers a lot of things in the body that we need, including exercise and muscle contractions and things like that. So ATP consists of an adenine molecule, a ribose molecule, and three linked phosphate molecules making up the TP part, so the triphosphate. And so when we are making ATP, which is what we do in bioenergetics, we're going to be typically taking an ADP molecule, so an adenosine diphosphate, so two phosphates, and we're going to be adding a phosphate to it, making it into ATP, so three phosphates. All right, so that process is called adenosine diphosphate phosphorylation, or ADP phosphorylation. And it's simply creating an ATP molecule from an ADP molecule and an inorganic phosphate. So adding the two together to make an ATP molecule. And of course, it's going to get more complicated than that, but this is the simplest explanation of how we create energy in the body. So let's go over the basic bioenergetic pathways. And this is just a real brief overview. We're going to be going over each of these in more detail in later videos within this series here. All right, so we're going to have some anaerobic processes. These are processes that don't require any oxygen. And we're going to have some aerobic processes, which are the ones that do require oxygen. So looking at the anaerobic processes first, we have immediate energy sources and fast energy sources. The immediate energy sources are going to be first the stored ATP. And what I mean by this is I'm just talking about the energy that was produced before that's just kind of sitting around the muscle waiting to be used. So we always have a little bit of ATP in our muscles or in the cells of the body that can be used on demand. And that's what I mean by stored ATP. So once the stored ATP starts to get used up, then we need to switch over to phosphocreatine as a way of creating the ATP. These together are our immediate energy sources because the stored ATP is the energy that's just sitting there in the phosphocreatine system is a really, really fast, basically instantaneous system for recreating that ATP. All right, and then we go to our fast energy sources. So now we're not quite as quick as being immediate, but still very, very quick. And this is going to be glycolysis. This is the system that breaks down carbohydrate, ending in this case, lactate if it's an anaerobic metabolism. And if we have aerobic metabolism, keeping up with metabolism, or keeping up with the energy needs, then we have glycolysis that ends in pyruvate. And we'll get into these details again in another video. I'll link to that video down in the description below this where we'll talk about glycolysis. And with the aerobic metabolism going through the glycolysis pathway, ending in pyruvate, that would then go through the aerobic pathways of the Krebs cycle, followed by the electron transport chain, which is also called oxidative phosphorylation. And there's different names for the Krebs cycle as well, the TCA cycle, the tricarboxylic acid cycle, citric acid cycle as well. All right, so this is carbohydrate breakdown. Again, these two are really the same processes. These are both glycolysis. It's just whether it happens quickly, it ends in lactate, and it can be fully anaerobic. If it happens more slowly, it produces pyruvate as its last product, and that goes into aerobic metabolism, which is a good thing. It makes a lot more energy that way. All right, so again, this is carbohydrates. Let's talk about fats. So the process of making energy from fats is beta oxidation, and this is going to be creating some molecules that will go also into the Krebs cycle, just like the aerobic version of glycolysis did. And so it goes into the Krebs cycle and then eventually into the electron transport chain, and it produces a lot of energy that way. Notice that we have carbohydrates, we have fats. We do not have protein listed on this slide. There's a reason for that, and let's go over that right now. Protein is just amino acids bound to nitrogen in such a way that basically our body can make use of it. And so from 1 gram of protein, we can liberate 4.1 kilocalories of energy if we need to. All right, so big caveat there is if we need to. If we overconsume protein, our bodies do not store protein as protein. We do have stored protein in our skeletal muscle and some other tissues and organs, but it's within the structure. We don't typically want to pull that protein out for anything, because that would mean breaking down your muscles, breaking down your organs. All right, so, but if we do overconsume protein, what we end up doing is we store it as body fat through lipogenesis, so just the creation of fat. When we use protein for energy, that process, or that it's sort of an umbrella term really, is called protein catabolism. It just means breaking down proteins. And is breaking it down into that nitrogen and those individual amino acids. The amino acids can be converted into various metabolic intermediaries that go into the citric acid cycle or the Krebs cycle. One of the common ones would be acetyl-CoA. Normally, protein catabolism contributes very little to our bioenergetic energy production. Somewhere between 2 and 10% is the typical range, and usually it's closer to that 2% range. In longer endurance type exercise, an hour, 2 hours, 3 hours, ultra marathons even, or in starvation type situations, it will contribute a greater and greater percentage of our total energy, up to about 10%. But again, normally we hover more closely to the 2% range. So protein minimally involved with the creation of energy. So what do we actually want to use protein for? We actually want to use protein in order to build up the tissues of the body, the skeletal muscle, right? The organs, heart, all that is very protein rich. The entire body is almost made of protein. Very few tissues aren't made of protein. And so we don't usually want to use protein for energy because we want to shuttle it towards maintaining and building and growing tissues. So again, protein, just to sort of drill this home, protein is not typically a major energy source during exercise or during rest. We do not want to use protein if we can avoid it. This was a very brief overview on bioenergetics, and we did get into protein here simply because I wanted to point out the fact that protein is not really a major part of bioenergetics. We'll have additional videos where I specifically go over carbohydrates and go over fats separately. And I'll also have a video talking about the immediate sources of energy when we start to exercise. Let's talk about the immediate bioenergetic energy sources. All right, so the first one, and it's not so much a pathway, but more of just a description of something that exists, and that is the stored ATP that's just sitting there waiting to be used in our muscles and various cells of the body. All right, so we always have a little bit of ATP sitting there ready to be used. So it's a very small amount. It's stored within the cytosol of the cells. So when we start to contract muscle, this is going to be the first ATP that we use. Again, just the stored ATP that was produced at a previous point in time. A typical person has about enough stored ATP in order to sustain intense exercise for somewhere between 1 and 3 seconds. And when we do this, anytime when we use ATP, regardless of the source, whether it's stored ATP or it comes from some other bioenergetic pathway that we're going to be talking about, the process of using the ATP is the same. It's always going to be ATP hydrolysis. ATP hydrolysis is just the term given to the process of breaking down ATP in order to release its energy. And so we can see this simple diagram here where we have the ATP molecule. This needs to be done in the presence of water, so we have a water molecule. So these two together, using the ATPase enzyme, is going to yield ADP, the inorganic phosphate, the energy that's liberated from pulling off that phosphate molecule. And there's also going to be a hydrogen ion, or basically an acid molecule, that's going to come from this. And this is actually going to be pretty important when we talk about the acidification that happens in the muscles and in the blood during intense anaerobic types of exercise. So we're not going to get into that in this video, but I'll try to put a link down in the description to that video once that video has been created. Once we've used up most of our stored ATP, we then need to start creating new ATP. And this is the first place where we really get into bioenergetics, so the process of creating energy. So phosphocreatine is that first energy system that we're going to use, especially with high intensity exercise. And it is an immediate energy system because it's so quick that our bodies aren't going to be able to sense the difference. We're not going to slow down. We're not going to fatigue going from stored ATP usage to creating ATP through phosphocreatine. And again, so it's an immediate energy system used to make new ATP from ADP, so broken down ATP molecules. This is going to happen in the cytosol of the cell. Does not require oxygen, so this is completely anaerobic. Our phosphocreatine stores are going to be able to fuel intense exercise for from the 3-second time point to about 15 seconds. So before 3 seconds, we're still using the stored ATP molecules, but starting around 1 to 3 seconds, somewhere in there, once we start blowing through some of that stored ATP, that's when the phosphocreatine takes over, and it's going to be able to fuel our exercise as a primary energy source up to about 15 seconds. At that point in time, we have to go away from the immediate energy sources and start going towards fast energy sources like glycolysis. Briefly looking at the chemical reaction here, we have our phosphocreatine molecule, we have an adenosine diphosphate molecule. Both those need to be present because we're going to be basically taking a phosphate off of the phosphocreatine, adding it to the ADP, making it ATP. And to do that, we need the enzyme creatine kinase. And again, we get the ATP and then the creatine molecule without its phosphate attached because it's been moved over to the ATP molecule. And this is a simple diagram of the phosphocreatine system. So once we blow through our stored ATP and our phosphocreatine stores, the next energy pathway is going to be glycolysis, a very fast pathway that uses carbohydrate. I'm going to put a link in the description below to the video where I'll be covering that. Glycolysis is our fast energy system. It's not an immediate energy system, so it's not the fastest, but it is our fast energy system. In this video, we're going to be talking about how we use carbohydrate in order to make energy in the body. So let's talk about carbohydrates and what it is real quick. So carbohydrate is a molecule that combines carbon, hydrogen, and oxygen in order to make a saccharide molecule. So when we have a carbohydrate that we use in our body for energy, we can liberate about 4.1 kilocalories per gram of carbohydrate within our body. And so the most important carbohydrate within our body is glucose. So this is what we're going to be focusing on here, and that's generally what you hear about and read about in textbooks when you're reading about bioenergetics and metabolism. All right, so glucose, again, the most important carbohydrate. It's a monosaccharide, which means it's a simple sugar. It only has a single saccharide molecule in it. And this is the carbohydrate that we are talking about when we say blood sugar. So blood sugar is glucose. This is also the primary energy source for the brain as well as the spinal cord. And when we do intense exercise, it is our body's primary energy source to fuel our muscles as well. So glucose is super, super important for normal physiological function, especially for exercise performance. All right, so glycolysis is the term for when we break down glucose in order to make energy from the glucose molecule. And it is going to be the primary source of energy for intense exercise from about 15 seconds out to about 2 minutes of continuous intense exercise. Another term that you should be aware of is something called gluconeogenesis. This is when we take various substances that are not glucose and we turn them into glucose. So the most commonly converted substances are going to be various amino acids, glycerol, and lactate. So all of these can be turned, as well as others, can be turned into glucose, again, through that process of gluconeogenesis, which primarily happens within the liver of the body. Our body does have the ability to store glucose, and when it does that, it stores it in a molecule called glycogen. So glycogen is nothing more than several glucose molecules that are bound together into a single large molecule. And so our primary places where we store glycogen is going to be muscle, especially skeletal muscle, and liver, within the liver's tissue. The process for creating glycogen is called glycogenesis, and the process for breaking down glycogen is called glycogenolysis. This diagram down here talks about how we can create and then also break down glycogen. So in this diagram, we have blood glucose coming past the cell, and this is depicting a skeletal muscle cell here. And the blood glucose can come into the cell. Once it's in the cell, using the enzyme hexokinase and using up an ATP molecule, we convert it to glucose 6-phosphate. Once it is turned into glucose 6-phosphate, the skeletal muscle does not have the ability to eject that or remove that glucose from the cell. So within skeletal muscle, we do not have the enzymes in order to get this back out of the cell. The liver does, skeletal muscle does not. Again, we're talking about skeletal muscle here though. So once we have the glucose 6-phosphate, it can go through this series of steps here, eventually turning into stored carbohydrate or stored glucose, again, as glycogen. Or in this case, specifically, we're talking about muscle glycogen, but it's the same as liver glycogen. The glycogen can be broken down and turned back into glucose 6-phosphate, which can then go down through the glycolysis pathway, continuing eventually making ATP. All right, so this top part right here is the same diagram we just showed when we were talking about glycogen. So this is that sort of cycle of making and breaking down glycogen right here. And here's that blood glucose coming in from the blood into the cell. All right, so whether it comes from blood glucose or comes from muscle glycogen stores, eventually it's going to turn into glucose 6-phosphate, and it's going to go down through all these steps here. This part that is in this purple box here is what is sometimes called the investment phase of glycolysis. And the reason it's called that is because we actually use energy up in order to go through these processes. Specifically right here, we use an energy molecule that was when we brought the glucose in and turned it into glucose 6-phosphate. And then we also use another ATP molecule here, going from fructose 6-phosphates to fructose 1,6-bisphosphate. And so these two steps do use up energy. This entire process of glycolysis, once it's no longer in the blood and actually in the cell, which would happen between this step here, it's going to happen in the cytosol, and it's going to happen anaerobically, so we don't need oxygen in order to do that. So that's both for the investment phase and what we're going to be talking about next, which is the harvest phase. In the harvest phase, the fructose 1,6-bisphosphate actually splits into two molecules, and when it does that, it goes down two parallel paths that are identical. Okay, so if you know one path, you know the other path. And that also means that for every molecule that is produced, so right here we're making an NAD molecule or an NADH molecule, we're actually making two of them because again, there's two pathways. So here's an NADH, here's an NADH. Right here we're making an ATP, here we're making another ATP. And then down here towards the bottom, here's an ATP, and yet again, here's another ATP. The final product in glycolysis is pyruvates in both of these parallel paths, assuming aerobic metabolism is able to keep up. All right, so looking at slow glycolysis, which is when aerobic metabolism can keep up, we end up making a net of 2 ATP, because remember, we did make 4 ATP, but in the investment phase, we used up 2, so it gives us a net of 2. We made 2 NADH molecules, and we made 2 pyruvate molecules that are eventually going to go into aerobic metabolism into the Krebs cycle. All right, so let's talk about, though, for a moment when we don't have slow glycolysis and when we actually have fast glycolysis. And what's the difference? Glycolysis can happen at various rates, so we can get what sometimes is called slow glycolysis and sometimes what is called fast glycolysis. All right, so depending on how quickly glycolysis is moving forward, we can either go slow and pump those 2 pyruvate molecules from each glucose molecule down this preferred path into the Krebs cycle, and then where it becomes acetyl-CoA, and is part of that Krebs cycle, and eventually we end up getting a lot more energy from it, which is why it's the preferred path. It goes down the aerobic pathway. If we completely fill up this pathway and we're still making a lot of pyruvate, which is what happens when glycolysis starts to run faster than aerobic metabolism, this pyruvate starts to spill over into a secondary pathway. And that secondary pathway is going to turn the pyruvate into lactate using the lactate dehydrogenase, and in the process, it actually uses up an NADH molecule, turning it back into an NAD molecule. All right, so if we do this, so if we go down this pathway here, we end up losing out on the pyruvate molecules that can go into the Krebs cycle. We also end up losing out on the NADH molecules that can go into the electron transport chain. And instead, we end up with lactate. Good thing is lactate, this pathway is reversible. So if metabolism slows down or bioenergetics slows down, glycolysis slows down, the lactate can be turned back into pyruvate, and you basically end up recreating the NADH, and the pyruvate can go down this preferred path at that point in time. But metabolism so basically exercise intensity needs to slow down enough for that to happen, or if you just stop and start resting, that will eventually happen to most of the lactate. It's important to note here that we're talking about fast and slow glycolysis here. These aren't necessarily distinct things. This is cell by cell specific. All right, so for instance, a fast switch muscle fiber, a type 2 muscle fiber, is going to have a weaker aerobic system than a slow twitch muscle fiber is going to. And so at the same rate of glycolysis, the fast twitch muscle fiber is probably going to be making lactate, but the slow twitch muscle fiber is probably going to be keeping it at the end product of glycolysis as pyruvate and sending it down the aerobic pathways. So different cells are going to produce lactate at different intensities of exercise. Let's go through a summary of glycolysis. So we start with glucose in the blood. It gets into the muscle cell or whatever other cell of the body you might be talking about, and it's going to produce 2 NADHs that are going to go on to the mitochondria to be used in aerobic metabolism. It's going to produce 2 ATP molecules that is going to be used immediately in order to keep exercise going. And it's going to produce 2 pyruvate molecules that are also going to go into the mitochondria in order to be used aerobically. So if we kind of zoom in on the mitochondria here, which is what we're showing down in this section, we have the pyruvate coming into the mitochondria, being turned into acetyl-CoA, goes through the Krebs cycle, produces some ATP, produces some NADHs, produces some FADH2 molecules, all of which the NADHs and the FADH2s are all going to go into the electron transport chain and produce more ATP molecules. So looking at this table here to kind of summarize what we're getting out of each part of the process, we have glycolysis that's going to produce 2 ATP molecules, 2 NADHs, and 2 pyruvate molecules. Again, assuming that aerobic metabolism is keeping up with glycolysis. We have pyruvate decarboxylation, which is a process I didn't really talk about a whole lot here, but it's actually this process of turning pyruvate into acetyl-CoA that's going to produce 2 NADHs right here, which is what's listed here under the other products of pyruvate decarboxylation. This is assuming that we're breaking down the whole glucose molecule, because remember, when we go through glycolysis, it actually splits into 2 pyruvates. So it goes through this process and this process twice. All right, so the Krebs cycle, again, going through it twice, we're going to produce 2 ATP molecules in total from 1 glucose molecule. Also 6 NADHs, 2 FADH2s. And then once the NADHs and FADH2 molecules get to the electron transport chain, that's where they get used up, and they produce 28 more ATP. So these additional 30 ATP molecules are only going to come from glycolysis when glycolysis is slow enough for aerobic metabolism to take in the pyruvate and use it to produce additional energy. So that's why we get so much more energy out of slower glycolysis per unit of glucose than we do at a faster rates of glycolysis. Here I just did a very brief overview of the Krebs cycle and the electron transport chain. I'm going to have an entire video that I'll link to in the description below talking about how these processes work and exactly what they do. I'll also put a link in the description below to a video talking about how fat metabolism is used for exercise and how much energy and how we can get energy out of that as well. In this video, we're going to be talking about the 2 parts of aerobic metabolism, so specifically the Krebs cycle and the electron transport chain. All right, so let's start with the Krebs cycle. Some other names for this would be the citric acid cycle in the tricarboxylic acid cycle, which this is often abbreviated TCA cycle. All of these, if you see them in textbooks or various sources online or other videos, they're all the same thing. They're all the Krebs cycle. All right, so the Krebs cycle is the starting point for the shared aerobic bioenergetic pathways, which is why we're starting with it in this video. In order for the Krebs cycle to work, we need to be fueling it with intermediaries, basically substances that it can use. All right, so there's various ways we can do this. We can do it through lots of different other energy substrates. All right, so let's talk about each of those right now. So glycolysis, which is the breakdown of glucose or carbohydrate, contributes to the Krebs cycle. This is what I have up here sort of highlighted in this green text and these green boxes and arrows. It contributes to it 2 pyruvate molecules per glucose molecule that's been broken down. So basically, that we're going to be sending a pyruvate molecule into here. It's going to be going through the process of pyruvate decarboxylation. In the process of doing that, we create an NADH molecule. We also produce a CO2, so this is the first time we're seeing CO2 being produced in bioenergetics outside of acid-base buffering that might happen in the blood. And we're also going to be producing an acetyl-CoA molecule, which is typically talked about as the starting place for the Krebs cycle, even though in reality there really is no starting place for the Krebs cycle, but it is the starting place for how carbohydrates contribute to the Krebs cycle. All right, so this is how we get intermediaries out of glycolysis and out of carbohydrates. If we are looking at fat, all right, so beta oxidation is the process of breaking down fat. It contributes to the Krebs cycle also through creating acetyl-CoA. So beta oxidation creates acetyl-CoA that does go into the Krebs cycle again there. Protein can also contribute to the Krebs cycle. The process of breaking down protein is called protein catabolism. Remember that protein is minimally involved in bioenergetics. Our body tries to conserve protein. It does not want to use protein in order to make energy. We want to use protein in order to build structures, things like muscle and different organs and tissues like that. So we don't typically want to do this, but we can when we need to. When the energy of the body starts to get low, protein will contribute more than it does when energy is prevalent in the body, but it's still, even when it's being at its highest, maybe 10% of total energy is going to come from protein. And so I didn't mark it in here. I did put a little note here about protein catabolism. Protein catabolism, depending on the amino acid being broken down and how it's being broken down, it can contribute to various different steps, including immediately into the acetyl-CoA step that the glycolysis and fat breakdown processes contributed to. So that was everything that gets put into the Krebs cycle. Let's talk about once the Krebs cycle is going, what's happening. So I think we first actually need to mention the fact that the Krebs cycle happens within the mitochondria. So all aerobic metabolism happens in the mitochondria. So the Krebs cycle requires oxygen to be present. Everything in the mitochondria, all the processes requires oxygen to be present, but the Krebs cycle itself does not use oxygen. When the oxygen is not present to keep the electron transport chain going, the Krebs cycle will back up, and that causes it to eventually stop or slow down dramatically. All right, so the Krebs cycle, though, goes through all these steps in this circle, which is why it's called a cycle. The Krebs cycle is going to be producing 3 NADH molecules, one here, and one here, through one full cycle of the Krebs cycle. If you are going to be starting with glycolysis and contributing pyruvate, it has to go through the pyruvate decarboxylation process in order to become acetyl-CoA. You also produce another NADH molecule right here, so that adds to the 3, so 3 plus 1, giving you 4 NADH molecules per pyruvate molecule that goes into the Krebs cycle. We also produce one FADH2 molecule right here, and we also produce one GTP molecule, which is kind of like ATP, but we don't use it so prevalently in the body. And it is actually immediately used in order to, it's converted down to GDP, releasing an inorganic phosphate, and in the process, we actually make an ATP from ADP. So from the Krebs cycle, we actually get one ATP molecule from this GTP molecule that was produced. And we also end up with 2 CO2 molecules being produced, one here and one here. A third one, again, if you're using glycolysis through pyruvate, the pyruvate decarboxylation, ending with this CO2 right here. So that would give you 3 CO2s if you're starting from the glycolysis angle into the Krebs cycle here. So I've talked a lot about NAD and FADH2, but I haven't really mentioned what it is. So let's do that now. So these two are going to be electron and hydrogen carrying molecules. The NAD stands for nicotinamide adenine dinucleotide, and when we make it into a high energy, we also add an extra hydrogen onto that. So that's when the NADH is the high energy version of that. And so the reason why it's high energy is because it has some electrons, actually, so it has 2 electrons, and in the process, it has a single hydrogen, and it also releases a hydrogen ion into the solution whenever we go from NAD to NADH. And so when we pump the NADH into the electron transport chain, which is where we're going soon with this video, we end up getting 2.5 ATP molecules per NADH molecule that is broken down. However, that is not the only way we can use NADH. We can also use it anaerobically. So when we're using a lot of anaerobic metabolism and glycolysis is running faster than aerobic metabolism can keep up, and we're making lactates, the production of lactate from pyruvate actually requires an NADH molecule in order to do that. So we break down one of the NADH molecules back into NAD in order to produce lactate from pyruvate. And so this is, a lot of people think of this as like a bad thing that we're making lactate, but actually lactate allows your glycolysis pathway to continue to function. Because if you just ended with pyruvate and we had a buildup of pyruvate because aerobic metabolism isn't keeping up with glycolysis would shut down, meaning we would have a significant decrease in our ability to produce energy in order to keep fueling exercise. All right, so, and this was NADH. Let's talk about the FAD or the FADH2. FAD stands for flavin adenine dinucleotide. In just like with NADH, FADH2 is the high energy version of FAD. When we take FAD and we add to it a couple hydrogen ions and 2 electrons, and we turn it into FADH2, that's actually the high energy version of FAD. So this is the version that's going to be going into the electron transport chain in order to make ATP, and we get 1.5 ATP molecules out of every FADH2 molecule that is broken down in the electron transport chain. So now let's look at the electron transport chain. Another name for this is oxidative phosphorylation. They're the same thing. So again, if you see that in a textbook, other videos, online, or other classes, we're talking about the same thing. So this also occurs in the mitochondria, and it is an oxidative process, so it's aerobic. Remember, all processes that happen in the mitochondria are aerobic processes. All right, so In this, we're going to be taking the NADHs and the FADH2 molecules that were produced in glycolysis and in the Krebs cycle, and we're actually going to be stripping off their electrons and stripping off their hydrogen ions in order to make this process go. So when we break down the NADHs and FADH2s, what we're actually going to be doing is we're going to be pumping the hydrogen ions from this inside the matrix or from these various molecules into this space here. So we have the mitochondria that has two membranes in it. So we have the outer membrane, which is what we have right here. And then we have this inner squiggly looking membrane here, which is what this membrane is right here, where all these complexes are embedded. All right, so these complexes, again, are going to be pumping the hydrogen ions into this intermembrane space, so the space between membranes. And by doing that, we are creating a gradient, a proton and hydrogen gradients, hydrogen being the proton, that will eventually be used in order to make ATP. So if you quickly kind of follow this process, we have an electron that comes off the NADH molecule, gets past the coenzyme Q, and passed eventually to this complex 3. We also have an electron coming off the FADH2 molecules going to coenzyme Q, again eventually being passed to complex 3. That electron then will go to cytochrome C that passes it to complex 4. So a lot of different steps here, but all it is handing off of the electron and in the process pumping hydrogen here, so here, and then also here into this intermembrane space. Then the electron eventually comes down here where it joins with oxygen and joins with some hydrogen ions and oxygen in order to produce water, so H2O. So the final electron acceptor here is going to be the oxygen. That's why I have this star here. It's a super important step. This is where oxygen finally becomes part of aerobic metabolism. So the oxygen becomes the final electron acceptor and the final hydrogen ion acceptor, creating water in the process. And what it did was created this large gradient here. So it's basically a battery within the mitochondria that can go through the ATP synthase complex. And so the hydrogen goes through this, kind of going down. Think of it like water going through a dam. And in the process, it is able to use this gradient to make ADP back into ATP, making all the energy that comes from the electron transport chain. Right, so from one glucose molecule, we end up with 28 ATP molecules from all this process here. We also end up with 12 water molecules. Hopefully before you watch this video, watch the glycolysis video where we talk about breaking down glucose, producing all these NADHs and FADH2s. But if you haven't, go back and watch that. I'll put a link in the description below for that. I'll also put a link in the description below to the video on how we use fat metabolism and break down fat in order to also get NADHs and FADH2s. And I'll also put a link in the description below to a video going over some of the various ways that the bioenergetic pathways are modulated to sort of speed up or slow down those pathways. Fat is actually where we get most of our energy during rest and also low intensity exercise. So let's go ahead and talk about fat and fat metabolism here. So what is fat? Fat is simply a molecule, a lipid molecule that combines glycerol and fatty acids into something our body can get energy out of. When we break down fat in our bodies, we can get 9.4 kilocalories per gram of fat. The process of creating fat is called lipogenesis, and we can do that from various different sources. So proteins, carbohydrates, and fat consumption can all lead to lipogenesis. So overconsumption of any of that does lead to fat production. Our body's primary way of storing fat is actually through triglycerides. So most of the fat in our body is triglycerides. In our primary places where we store it is going to be our muscle and adipose tissue, which is what's listed down here. So we get somewhere around 60,000 to 100,000 kilocalories worth of fat in our adipose tissue in just a typical young, healthy man. And so we get around 3,000 kilocalories of fat that is sort of spread throughout our muscles and stored within our muscles, also again in a typical young man. And as I already mentioned, the triglyceride molecule is where we get most of our energy for rest and light intensity exercise. There are other uses for fats and lipids besides just bioenergetics and production of energy. The production of steroid hormones, which is hormones that are derived from cholesterol lipids, is going to also require fats or lipids. And so examples of that would be testosterone, estrogen, progesterone. There are many more as well. And also, all the cells of our body have a bilayer lipid membrane, so we need lipids and fats in our body in order to produce those. The breakdown of triglycerides in order to get glycerol in 3 fatty acid molecules is called lipolysis. So anytime we want to use the triglyceride molecule in order to make energy or do anything else with it, we need to break it down again through lipolysis. So just to quickly look over this one more time, here's an example of a diagram of a triglyceride. We have the glycerol in our 3 fatty acids attached to it. We need to have some water, so 3 water molecules in order for this to happen. We have the enzyme lipase, and that's going to help split this up into, again, the glycerol and the 3 fatty acid molecules. The glycerol molecule, this one here, that's the structural backbone of the triglyceride. It's not a waste product. We can use it. Obviously, we can use it to reproduce triglycerides. We can use the glycerol molecule in glycolysis in place of glyceraldehyde 3-phosphate in order to create 19 ATP molecules from a single glycerol molecule. So it can be used for energy directly. It can also be converted into glucose through gluconeogenesis that will create a glucose molecule that can then go through glycolysis that way. So again, the glycerol molecule is not a waste product. We can use it for various different things, including energy production. The fatty acid molecules, these are the single lipid units that are energy-containing lipid units within the body. So these are eventually going to go into beta oxidation, where it breaks down the fatty acids within the mitochondria into acetyl-CoA in order to make ATP. This slide overviews the breakdown of fatty acids through aerobic metabolism. So fatty acids are, first of all, transported through the blood bound to albumin, primarily bound to albumin. And it's eventually brought into the cell, shown here in this diagram, through specialized transport proteins that allow the fatty acid to come in. Once the fatty acid is inside the cell, it is converted into its activated form of fatty acetyl-CoA in an energy-intensive process that actually breaks down one ATP molecule and turns it into ADP, using that energy again to make the fatty acetyl-CoA. The fatty acetyl-CoA can then enter the mitochondria. So this is where we're finally getting into the mitochondria, and it enters it using the carnitine shuttle. Once the fatty acetyl-CoA molecule gets into the beta oxidation cycle, it's going to pull off 2 carbon molecules per cycle and convert those into acetyl-CoA, where it can be thrown into the Krebs cycle and used during aerobic metabolism. I've already done a video talking about the Krebs cycle and electron transport chain aerobic metabolism. I'll put a link in the description below this video to that video, in case you haven't seen that already. All right, but this process, this beta oxidation process of removing 2 carbon molecules, is going to keep happening over and over and over again until there are no carbon molecules left to remove from the fatty acetyl-CoA molecule. And there can be several carbon molecules in one of these molecules. So this process can happen several times over, producing a lot of energy eventually. Once the acetyl-CoA gets into the Krebs cycle and it goes through the normal Krebs cycle process, we're going to end up getting NADHs and FADH2s. The NADHs and FADH2 molecules produced in the Krebs cycle will then make their way into the electron transport chain, where they get used up to produce a lot of ATP. All right, so much more ATP from fat than what you're going to get from carbohydrate. So I'll put a link in the description below to that video where I described the Krebs cycle and the electron transport chain in a lot more detail, which is the 2 aerobic pathways that beta oxidation is feeding into. I'll also put a link in the description below to a video where I talk about glucose breakdown, because these 2 things, the fat and the glucose, are our primary energy sources that our body uses, especially during exercise, where there's a sort of progressive shift from fat to glucose. So what is an enzyme? An enzyme is a molecule that lowers the energy required to make some sort of reaction take place in the body. The reaction could take place without the enzyme, but it would be much more difficult, so it wouldn't happen nearly as often. All right, so in this diagram, this gray molecule is our enzyme, and our enzyme becomes active once the substrate specific to it binds. All right, so here's our example substrate molecule. Notice that the shape of them fit together. So when they come together into the enzyme substrate complex, that is when we have our activated enzyme, and it's going to act on that substrate to do some sort of action. So let's use a metaphor to help us here. All right, so here we have this person who's trying to get down to the waters and maybe fish or swim or do whatever this person's trying to do down at the water. And there's this wall in the way. And in the wall is a door with a keyhole, and this person may or may not have the key. All right, so the key is the substrate, and the enzyme is the keyhole here. So if we do not have the key, the person has to go up and above and up and over the wall in order to get to the water. So a lot of energy is used to get over this wall. When we have the key, we can go right through the door. So we just open the door. A little bit of energy is used, but not nearly as much energy. All right, so how does this work in the body in real life? So getting beyond the metaphor here. All right, so we have energy that has to be put into the reaction or in order to activate it and make it go forward. And so this is our activation energy. Everything above this green line here in this red space is our activation energy. So we want to minimize our activation energy to make the process as efficient as possible. Once the reaction is activated, it's going to go all the way. It's going to break down the substrate into whatever it's going to be broken down into, or combine the substrate with another substrate into whatever it's going to be combined into. In this process, we're talking about energy, so typically it's going to be broken down though. If we have the enzyme present, the activation energy, again, is much smaller in order to get this process to carry out breaking down the substrate. And so if you were to look at all this, so we have our loss energy, which is our sort of activation investment energy. Our loss energy is much larger without the enzyme than it is with the enzyme. The amount of energy gained from breaking down the molecule is the same, so we have the same amount of gained energy. However, the gained energy is not what necessarily is important, it's the net energy. So removing the lost energy from the gained energy gives you your net. So without the enzyme present, we get some energy. With the enzyme present, we get quite a bit more energy, net energy out of breaking down this substrate. Because again, you don't need to have as much investment energy in order to get the substrate to break down. Most enzymes are going to end in the letters ASE, so anytime you hear something ending in ASE, it's probably an enzyme. What are the factors that regulate enzymes? There are several out there, but two of the big ones are temperature and pH, or acidity. So as the body's temperature goes up, the enzymes, most enzymes are going to increase their activity. This is something that happens with exercise. So as our bodies heat up, our enzymes become more active. pH, as the pH reduces, so as we become more acidic, which also happens during exercise, typically enzymes actually become less active this way. So the heat produced during exercise typically increases enzyme activity, where the acidity that's produced, or the extra acid that's produced with exercise, is typically going to decrease the enzyme activity. And I just want to one more time emphasize that the enzyme and substrate need to fit together. It's a lock and key style model. If they don't fit together properly, their physical form is not the proper form in order to get and combine together, then it's not going to happen. So if anything changes the shape of these enzymes, so any of these various modulating factors can possibly change the shape of the enzyme or the substrate, it's going to make it so it's harder for these two, the substrate and the enzyme, in order to get together and have their action. So at the beginning of this video, I mentioned how enzymes actually control the rate of bioenergetics. And so we have what we call rate-limiting enzymes. These are the enzymes in each bioenergetic pathway that is going to be either the slowest or the most scarce. So there's not enough of it essentially to go around in order to make the pathway go quickly. And so whichever is the The slowest or the most scarce enzyme is typically going to be your rate-limiting enzyme, and it is the enzyme that slows down the metabolic pathway and controls how quickly that pathway moves forward. So if you were to improve the rate-limiting enzyme, you'd actually speed up the pathway so you can get more energy out of that pathway. All right, so what are some various modulators of these enzymes besides just temperature and pH? ATP, ADP, so the broken down version of ATP, and also the inorganic phosphate coming off of the ATP when it's broken down, those are all very common modulators. Typically, when you have high levels of ATP, it's going to inhibit the bioenergetic pathways and the enzymes that are the rate-limiting enzymes. When you have low amounts of ATP, meaning your body needs to produce it, that's going to speed up the pathways and speed up these rate-limiting enzymes. Likewise, if you have a lot of the breakdown product of ATP, so again the ADP and the inorganic phosphate, it's also going to stimulate these enzymes. A lot of the enzymes are going to work through a negative feedback system. So basically, as you produce and stockpile the end products of those processes, you're going to slow down the process. It's going to feed back and give a negative feedback on the process to slow down because you have a lot of the product anyways. You have a stockpile of the product, so you don't need to make more. So it kind of makes sense that you use a negative feedback system to help modulate the bioenergetic pathways and the rate-limiting enzyme speed. All right, so what are the various rate-limiting enzymes and specific stimulators and inhibitors of the various metabolic processes? So phosphocreatine's rate-limiting enzyme is creatine kinase, which is actually the only enzyme in the phosphocreatine pathway. It's going to be stimulated by ADP, and it's going to be inhibited by ATP, just like I mentioned already. Glycolysis, the rate-limiting enzyme, is the phosphofructokinase, and it's going to be stimulated by AMP, which is also a breakdown product of ATP breakdown. So it's basically when you pull off two of the phosphates, so you get adenosine monophosphate, so a single phosphate, the ADP, the inorganic phosphate, or a high pH. Inhibitors are going to be ATP, phosphocreatine, citrate, and low pH. The Krebs cycle, the rate-limiting enzyme is isocitrate dehydrogenase. The stimulators are ADP, calcium, NAD. The inhibitors are ATP and NADH. In the electron transport chain, the rate-limiting enzyme is cytochrome oxidase. The stimulators are ADP and inorganic phosphate, and the primary inhibitor is ATP. There's additional molecules that likely stimulate and inhibit each of these processes, but these are the big ones that I mentioned here. Hopefully this video on enzymes and bioenergetics was helpful to you. I'll put a link in the description below to a summary video on bioenergetics, and I'll also put links in the description below on a specific video on carbohydrate breakdown and a specific video on fat breakdown and a specific video on aerobic metabolism. In this video, we're going to be talking about how much energy we have in carbohydrates and fats within the body. And we're also going to be going over a sort of summary overview, global style overview on bioenergetics. All right, so let's get into talking about our energy stores in the body. First, though, I want to remind you that our body prefers not to use protein for the production of energy. Our body likes to use protein in order to build things like muscle and organ tissue. That we can use it for energy, but we tend not to, so we're not going to talk about that on this slide. All right, so we have our carbohydrate storage locations, so various fluids in the body. For instance, the blood, we have blood glucose. We have our liver that stores around 451 kilocalories worth of energy in carbohydrate. We also have our muscles that stores about 2,050 calories worth of carbohydrate. In the liver and the muscle stored carbohydrates, it's going to be in the form of glycogen, where the fluids and primarily blood is going to be in the storage form. Well, not really a storage form, but it's going to be in glucose. Looking at fats, we have fats stored in the muscle. We have around 1,513 calories of fat stored in the muscles around the body. So roughly, this is going to be, all this is based on 65 kilogram adults with about 12% body fat. So obviously how much fat you store is going to go up if your body fat percentage goes up. All right, and we also have subcutaneous and visceral fat that is stored in the body, and we have a 73,320 calories on average stored in this subcutaneous and visceral fat. So far more than we have stored of anything else. Notice I actually had to put a break in the axis here because I couldn't possibly show this bar while also showing these other smaller bars because the y-axis would be so large that these would look like nothing. All right, so if we put all the carbohydrate sources together, so all of these, and all the fat sources together, so these two, we end up with around 2,563 kilocalories worth of carbohydrates stored in the body, and around 74,833 kilocalories worth of fat stored in the body. So we have far, far more fat than we have carbohydrates in our body that we can use for energy. So talking more about these various sources of energy, again, when we consume food, we can get fats, carbohydrates, and proteins from that food that can eventually be used for energy. We can also use alcohol for energy, but it's not something our body normally does, and we can't do a whole lot of it. So it's not on this slide. It's also something that for health reasons and for exercise performance reasons, you generally don't want to consume alcohol. So we're not going to talk about alcohol here. So when you consume these foodstuffs, though, it has to go through digestion. And so our dietary fats, think triglycerides, get broken into free fatty acids and glycerols. The free fatty acids can either go through lipogenesis to be stored as adipose tissue, which is stored back as triglycerides, or we can go the other way and we can go through the stored adipose tissue and the triglycerides can go through lipolysis to become fatty acids. So this is a cycle that can go both ways. And when you have fatty acids, they can go through beta oxidation, the Krebs cycle, and the electron transport chain in order to produce energy. The glycerol molecule that comes from the breakdown of triglycerides, either through consumption in our diet or when we break down the triglycerides in our adipose tissue producing fatty acids, and we also produce glycerol this way, that can go through gluconeogenesis to produce glucose, or it can go directly into glycolysis, then the Krebs cycle, then the electron transport chain to produce ATP. All right, so that's how our body uses the fats we eat. All right, so how does our body use the carbohydrates we eat? For the most part, we either bring it in as glucose, or we convert it to glucose in the body, all the carbohydrates. And so glucose, if it's in excess, can go through lipogenesis to produce fat tissue. So if the glucose isn't being consumed in excess, we can go through the process of glycogenesis in order to produce glycogen, which is the storage form of glucose stored in the muscles and the liver. We can also break down that glycogen through glycogenolysis back into glucose that can be used directly through glycolysis, the Krebs cycle, electron transport chain, in order to produce ATP. And then the last major foodstuff that we bring in through our digestion is the dietary proteins, which would become amino acids. Those amino acids can, when needed, go through gluconeogenesis to produce glucose. Our body doesn't want to do this. Again, we don't want to use protein for energy, but we can if we need to. We can also go through this pathway here, breaking down amino acids through protein catabolism, and produce ATP through various different pathways. So it has a lot of different places where it can fit in that way. We can, ideally though, what we would want to do is we would want to store the protein. So we'll go through protein anabolism in order to store the protein as skeletal muscle or organ tissue. But again, that can be broken back down through protein catabolism into amino acids, and again, used through this sort of pathway, various pathways to make ATP. If you overconsume amino acids or protein, we are going to just shove the amino acids through lipogenesis, producing adipose tissue. If you overconsume any of these, so fats, carbohydrates, or proteins, you're going to make fat. So we don't want to overconsume any of these because, again, it's just going to be turned into fat. This is a very brief overview of some of the various bioenergetic pathways and how it connects to the foods we eat. I have videos already talking about how we use glycolysis and how we break down carbohydrates. We have also videos on the breakdown of fats. I'll put links to each of those videos in the description below this video.